raised that yesterday. Hi, I'm Jia Yingtan, Scientific Editor at Cell, and I'm here for the 80s Cold Spring Harbor Symposium. This year's topic is 21st Century Genetics, Genes at Work. It's really a great pleasure of mine to have Dr. Davor Solter join me here and for the interview. And Dr. Solter has really made a great contribution to embryonic development and from pretty much all different aspects. So I was curious, to begin with, how do you get interested in this topic to begin with in your early stage?
Probably by accident. So I was teaching anatomy in Zagreb in medical school, and then I realized that one doesn't really do much experiments in anatomy, especially not in gross anatomy. So I went to, on advice of a friend, to a department of biology, and they were doing mammalian development actually in mid-60s, which was very unusual, certainly very unusual in Croatia and even unusual in the world, because mammalian embryos were considered as too small and too difficult and just not thing to do. And it was the very beginning of molecular biology, ultracentrifuges and stuff like that. And obviously for that you need buckets of embryos, so everything was done on sea urchins and xenopus. But I started doing mouse development, actually early mouse development, by first asking what would happen if you take early post-implantation embryo, so the early gastrula of a mouse, and transfer it to extrauterine site. And to my surprise, what I expected that there would be similar experiments were kind of hinted before, and they were done in rats. So one expected that it would be a very small kind of growth with very mixed tissues, but surprisingly, the things which grew, the tumors which grew, were actually very big, and after months or so, they would be almost as big as a mouse, and obviously would kill a mouse eventually. So the embryo just transplanted under the kidney capsule would turn into a malignant teratocarcinoma.
Right.
And that was my first beginning of interest into. We didn't know that embryonic stem cell existed even at that time, but slowly we realized that if you can make tumors out of embryos, then you can maybe make even cell line out of embryos.
Right.
And that's how we started. And then I slowly moved back into pre-implantation embryos and going back and forth for the last 40 years.
I see.
So that's how it started.
I see. Very fascinating. So lots of really great, I think, discoveries coming from all of the endeavors that from you and, of course, your lab. And what I think one of the most, people definitely honor that a lot, and it's really you're one of the pioneers that discovering the whole concept of imprinting. I think that really sort of changed at the time the way people think about this process. So over the years, what's your view in terms of the concept of imprinting has been evolving, if any, and what's sort of the way that people see it now?
Well, the time when we started those experiments in early 80s, 81, 82, obviously that I always use imprinting as an example of real discovery. So, you have a great scientific findings like finding the sequence of DNA, structure of DNA, which always the people mention. But was that a real discovery? The DNA was there, and we knew that it has a structure. I hope nobody in Cold Spring Harbor will object to that. And so you just had to find a structure. However, with imprinting, the reason why we started doing nuclear transfers were totally different. And then the reason why we started doing only male or female pronuclear structure was again completely different. We wanted to actually explain why parthenogenesis in mammals doesn't work. And we were fully expecting that embryo to which you transfer two male or two female pronuclei will develop perfectly fine because they were normal diploid embryos which have all the necessary chromosomes, and the problems which were invoked always as a cause of parthenogenetic failure were eliminated by doing nuclear transfers. Because we eliminated the problem, the zygotes was normally fertilized, so any contribution of sperm was already there. So, to our surprise, they never developed. And so it took us, Jim McGrath and me, probably 2-3 years to do those experiments until we were really convinced that these embryos are not going to develop and that there must be something different between the male and the female genome. And that was the beginning of imprinting. So result completely unexpected, because none of the classical Mendelian genetics would suggest that something like that should exist. And then you always are not sure where is the problem. You even at that time we were kind of fairly cautious. It is most likely that these are related somehow to the gene expression. But it was no evidence for it, and until the first imprinted gene was found almost 10 years later, that was the first time that we could really breathe the sigh of relief and say, yes, imprinting is truly genetically based mechanism. And today, and in a way, interestingly, the imprinting started this interest in epigenetics, which is now so prevalent and so modified, and I sort of flatter myself that imprinting actually started people thinking seriously about epigenetics again. But if you think how much we learn about, let's say, strictly imprinting subject from early 80s till now, so that's 30 years, there are still some basic, truly basic questions which we don't know, and I don't even know how to get to understand them. Namely, how does oocyte or how during oogenesis or spermatogenesis, how are these genes elected to be imprinted? We quite always say, sure, they become methylated, but methylation is an obviously later step which makes imprinting firmly established. What is the first sign, or what is the how the choice was memory? I mean, how does the oocyte receive these genes in completely native state and then treat some of them differently than all others? And how is this selected? We still don't know. And somehow, since we don't know how to even approach it, everybody works on how the imprinting is maintained and protected and saved and so on. But nobody quite wants to go into and try to see what is the first thing which made the imprinted genes different than others.
No, I think the initiation is definitely a key issue, and then that goes to not just the mechanism of maintaining the whole process, it's more of the principle of why to do this to begin with.
Well, the why to do it is also completely open, because the imprinting is certainly in the way what we understand is certainly restricted to mammals. It's probably present in some form in flowering plants, and it's not present in any other organism. So was imprinting invented as a mammalian approach to development? The evolution always poses a problem, but evolution never or rarely goes into to find an optimal solution. It finds a solution, and then we are stuck with that solution forever. And reproduction of mammals had faced a major problem, namely how to first start having embryos which are entirely dependent on another organism. They have to develop in another organism completely throughout their entire life. So they normally embryo of non-mammals, their first job is to appoint different portion of the embryo in different places.
Yep.
Mammals, their first job is make a trophectoderm.
Yep.
If they don't make a trophectoderm and don't succeed, the embryo will fail.
Yep.
And so, is it imprinting a way? Was it imprinting necessary for
I see.
developing trophectoderm?
I
That's
see.
one possibility. Is it imprinting? There, the favorite and most popular kind of theory is that balances maternal and embryonic conflict, which I don't know. I always feel that fetus and mother cooperate and they have a very rarely opposite interest, but it's a possibility.
Yeah, it's a very dynamic and very interesting relationship from the very beginning. Yeah, so I mean, you are also like one of, I mean, nowadays, like people talk about like stem cells, embryonic stem cells, and reprogramming all the time from all different aspects, and you're actually one of the really early scientists who started working on this program. And I think right now that with all those different ways of reprogramming, and people start to even think about whether it's therapeutic applicable, and where we go from there with, you know, with combination of transcription factor to small molecules and other things. So how do you see the field evolves in terms of where it goes?
Well, you can say there are two possible aspects. One was when the embryonic stem cells were derived in mouse.
Yep.
We all, very small group of people, thought they are very interesting cells and many interesting things will happen. And it didn't have any general interest or publicity or anything. And then when the human ESLs appeared, which isolated by my graduate student Jeremy Thompson, who then left, what happened is that somehow this happened, and coincidentally in the same time Dolly happened.
Yes.
And Dolly also created suddenly interest like nobody thought of doing nuclear transfer and cloning before, which was going on also for 10, 15 years, but Dolly suddenly attracted public imagination, as did the stem cells, human embryonic stem cells. And they were immediately seen as some panacea, that they will cure everything and change old age. I remember front page of the New York Times magazine which say racing toward immortality, and so on. So today I would think it's obviously that for purely scientific reasons we have a very little use for human embryonic stem cells. Most of these things can be done in mouse. However, in the therapeutic application of human embryonic stem cells, which had to be derived by therapeutic cloning, was mired for a long time in this discussion. Should we make embryos and then kill embryos just to cure people and so on? So now finally, at least this ethical mess was resolved by the induced pluripotent stem cells. And now the only question to my mind is the theoretical potential of induced pluripotent stem cells actually going to be realistic? Are we really going to be able to cure people? And to my mind, the questions of how to best reprogram, and are the iPS cells similar to embryonic stem cells, and what is exactly their differentiation potential, are all in a sense irrelevant. The question is, can you make from you or from me, can you take a cell and make it into another cell which will deal with the disease which you or I have? And it's totally irrelevant whether these cells, which let's say you need it to replace your cardiomyocytes, it's totally irrelevant whether this cell can also make neurons, because you don't need neurons. And if I need neurons, it's totally irrelevant whether they can make cardiomyocytes. So I think that the research is unfortunately not quite concentrating on the crucial issues, how to make it therapeutically feasible. How are we going to simply induce correct differentiation? Because obviously nobody is going to inject iPS cells which are going to make tumors. What are the clinical trials which we will have to make before we start injecting those cells into people. How we are going to send those cells into the right place?
Right.
What is the issue going to be if you realize this cell is not in a way normal? It had a strange history. It has been, let's say, it has been fibroblast. Now we treated it somehow, changed into iPS cells. Then we treated it again, changed into cardiomyocytes. Is this totally destabilized the genome? So you inject these cells into heart, and these cells, okay, some are making cardiomyocytes, some are making suddenly bone.
Yeah.
And now you have this misdifferentiation in a place which you totally don't want it. And also, how we can ever be sure that they will not make tumors? With all these experiments which we do in mice, we inject and they are fine and nothing happens. But mice live 1 year,
Right.
and people publish after 2 months.
Yeah.
We are going to treat children with those cells. I mean, what is the safety issues? And all those seem to me not quite attracting as much attention as they should,
I see.
and they will be all have to be dealt with, because this is totally new paradigm. I mean,
Right.
all the classical way of testing drugs and medical procedure, they are all have an element that if something bad starts happening, you stop.
Right. Yes.
But you inject the cells, and
Yeah.
there is no way getting them out again.
Yeah.
And so I see a lot of problems before. The potential is great, but the issues are also very important and not quite resolved.
Right. So it definitely still need to learn a whole lot, and also have an entire sort of probably like criteria laid out of how to move forward. What's the best way to evaluate the results, and what's the best way to really make it like step by step, like close to application.
Yeah.
Yep. Well, it's great to discuss this with you. I really learned
No
a lot.
problem.
I really enjoyed the conversation.
Thank you
so
very
much.
Thank you.
Yep.
Bye.